All right, welcome to the annual business meeting of the Tucson Herp Society. Thank you very much for coming. I hope you guys can hear me back there. Yep. Oh, good. Thank you. All right. So, the Tucson Herpetological Society, this is rounds out another successful year as organization which is dedicated to the conservation research and education concerning the amphibians and reptiles of Arizona and Mexico. We are situated in the Sonoran Desert, which is the traditional homelands of the Tohono and many other indigenous groups. I work at the Desert Laboratory on Tumamak Hill, which is also known as Chemamak Doag, or Hill of the Horned Lizard. And from there, I can look out across the Tucson Valley and I can look at Push Peak, which is there on the very left. And on the west side of Push Peak, on the west wall, you see why the Catalina Mountains are known as Baba Dak Doag, or Toad Sitting Mountain. There's an outline of a giant toad. And there's a Paiute story, which is southern Nevada, of a woman who bewitched her father and then turned into a toad, went underground, and popped up in the Catalina Mountains. And the story transcends many indigenous groups along the Colorado River and explains that this toad appeared somewhere between Tucson and Phoenix. And so this toad was once a witch. Let's see. So before we move on, I'd like to recognize two of our members who are no longer with us, Warren Savory and Royce Ballinger. They passed away this year. Warren was a former member of the THS board. He was the world expert on sulfugids, or sun spiders, and he sadly passed away in Guatemala. And Royce Ballinger also passed away last year. He did a lot of work with lizards and in Mexico. We have some of his artwork here from his estate, which he has donated, his family donated for us to fundraise with. So we have some items in the silent auction, which I hope you will bid on, and some items for sale, including a very unusual, unique candle holder from Oaxaca, which is a big mother turtle with her babies all around her. So the Tucson Herpetological Society is part of a coalition of organizations and individuals which is pushing for what would be the United States' second urban national wildlife refuge. The Santa Cruz River between Nogales and north of Marana. There are various parcels of land which county governments and private property owners have offered up to potentially be part of a series of properties that would be the Santa Cruz River Urban National Wildlife Refuge. So we are on that coalition. And on Thursday, the head of the U.S. Fish and Wildlife Service will be at the Canoa Ranch signing wildlife partnership, which is the first step towards being designated a urban national wildlife refuge. If you want more information, just see me after the presentation. So this year, these are our photo covers that I'm very proud of. Last year, we moved to feature a photo or a couple of photos on the cover of our Sonoran Herpetologist. And Howard Clark, our chief editor, has done an excellent job curating and editing the Sonoran Herpetologist for us. So these are the three from this year so far. The next Sonoran Herpetologist will land in your inboxes around mid-December. So this is a list of outreach events that we've done this year, and that's the total number of people, roughly, that we have reached in some capacity, whether in a classroom or a public event like the Tucson Festival of Books or the Tucson Reptile and Amphibian Show. I have to thank Patrick for heading up the Speakers Bureau and outreach events. He has really put in a lot of work to help spread awareness on our behalf of amphibians and reptiles. On top of that, he has a child that just turned 1 year old. So, and he's a full-time firefighter. So I think he might need some more responsibilities. So if you think of any, just let him know, okay? So thank you, Patrick, very much. And while I'm thanking people, I would like to thank Maggie and Lee Oler and Karen Watson and Ryan Perry and Hill Johnson for, in some capacity, helping put this event together that we're all at tonight. So it takes a lot of work and coordination ahead of time. And so thank you very much for doing that. When you donate to the Tucson Herpetological Society, you help us do lots of things. Print out coloring books, buy display cages, and do field trips, which we're still working on to try and make more of a regular thing. Our first, I think our first and only field trip of the year was in May to the Armendariz and Ladder ranches in New Mexico. We visited the only U.S. Population, captive assurance population of bolson tortoises. So the bolson tortoise is our largest terrestrial reptile, and its closest cousin is the Florida gopher tortoise. But this tortoise was found throughout much of the Chihuahuan Desert, and there's an effort underway to reintroduce this tortoise back to some of its historic range in the United States and in Mexico. And we've pledged $1,000 to this project for genetic testing of the individuals in this assurance colony. We had a great time, and I don't know who that is, but we did meet Gertie, the founding matriarch on that trip. And last year, we closed the Mexican Tortoise Fund. So the Mexican Tortoise Fund was a fund of money that did not belong to the Tucson Herp Society, but was held for biologists that were working with tortoises in Mexico and still are working with tortoises in Mexico. But we closed out this fund where they would hold money to do their conservation projects. Well, the last of that fund went to help secure, support work towards creating a federal reserve for a very unique ecosystem on the southwestern coast of Sonora called El Pitayal. It's basically an Oregon pipe cactus forest, and there's a population of tortoises that live in this coastal thorn scrub amongst these giant cacti. And so as we speak, I think it's the last day of the expedition to catalog flora and fauna and understand the status of the tortoise population there. So we recently provided them with $1,000 to help fulfill this expedition. We have the Charles Lowe Herp Research Fund, which this year we didn't get around to awarding because of timing, and we need people to help organize that review of projects, proposals. But we did award $4,000 to 2 projects from the Sonora Desert Toad Fund. I know I've said this many times, but for the sake of the recording and this being on YouTube, the Sonora Desert Toad Fund was established because the Sonora Desert Toad has been more popular because it's a source of 5-MeO-DMT, a very powerful psychedelic the most powerful psychedelic known to humans is produced by the Sonoran Desert toad. And why is that? We don't know. It's a very human-centric question to ask, actually. But here are the projects that we funded from that. And one way that we raise money is through merchandise. I have plenty of that here. At this table, there's merchandise items and some art. Actually, we have 2 framed pieces with artwork from Dennis Caldwell. Dennis designed our logo. He's an accomplished artist as well as a wetlands restorationist, but we have some really good deals on his art, which came to us from Royce Ballinger. And of course, we have toad merchandise, which can be found online, which supports our Sonora Desert Toad Fund. Many of our members have written excellent books. The Mojave Rattlesnake book was written by Mike Cardwell, which is here in our silent auction, a signed copy. Snakes of Arizona and all of the subsequent titles that'll come out are many of our members are authors in that series. If you have photographs or observations to contribute to the follow-up volumes, go to this website. So if you're looking for an alternative to Twitter, there's a new platform much like it called Blue Sky, and I just opened up an account for the Tucson Herp Society. So from here, I will hand it off to Maggie.
Close to me. Okay, so guess what? I have to talk about money because I'm the treasurer. This is, well, before I talk about money, you all know that you've got a nice little packet of cards to take home, thanks to Lee Oler. And some of you will remember last year we were giving these away, ornaments, really cute. Well, we've got a few left. You see those red boxes behind you by the wall there? Please, if you would like to have one or have another one, please pick it up. Stay there. So this is a gross overall summary. We have money, but you will notice, please, that our expenses have exceeded our income this year. This is a slightly larger breakdown of it, and there's a copy of this on each table if anyone would care to peruse it a little bit longer. I will point out to you that our memberships are struggling along. There's a few lapsed members who need to renew, so if you know anybody like that, you can nudge them. If you don't, I will probably be doing that soon. Our merchandise sales have been doing quite well, because we've been to some large events, and also because not only do we have the t-shirts and the caps, Robert has arranged for us to have a lot of books. We've had books donated to us, very fine books, and we've been able to fundraise with those. Donations at the moment are pathetic. We need money. We would like to do another Jocko Award this year. We need some donations. We bought a lot of merchandise. We probably won't have to buy much more. We would like to continue to give a lot of those grants and awards and sponsorships, and we really need funding for that. Operations supports the day-to-day stuff, but it also supports all the work that Patrick's been doing on outreach. The wild apricot is high because we do a 2-year contract with them. But the bottom line is, well, we need money. Our expenses exceed our income. Our income, just so as you know, over $9,000 are restricted funds, which means they are donated for a specific purpose, and we must then spend them for that purpose, whether it's Jocko or the Toad Fund or the low grant fund. I've already told you Wild Apricot was a 2-year renewal. The annual meeting isn't even in our expenses yet. And we do need to work on increasing our online merchandise sales, renewing lapsed members, and yes, trust me, you're going to get an end of the year donation appeal, but if you'd like to start tonight, this little froggy is going to be over there on the table. If you've got a little loose change in your pocket, you can stuff it in there, and that would be really fantastic. I think I've said everything I need to say. Any questions? Well, any questions, I'll be here and you can take a look at the budget. And now we should probably go ahead and move along.
Thank you, Maggie. And I realized as you were talking that I forgot to go over the fact that we awarded Larry Jones the Jarco Conservation Award this year as well for all of his outstanding contributions. Larry Lizard Lover Jones, as he's known affectionately. The Jarco Conservation Award, if you are awarded that award, you get a honorary lifetime membership, a plaque, a small ceremony, and I think it's $500 or whatever you want to do. And secondly, we would like to revisit the Bolson tortoise ranches, the Armandaris and Ladder ranches, not just to visit the amazing tortoises there, but there seems to be a very unusual population of horned lizards there that don't quite fit the appearance of what occurs there. Either it's cornutum or not platyrhynus, the one that looks like a little stone. So there appears to be possibly a hybrid population of horned lizards in this area where there's a bunch of lava fields. So now you know what we do with money and what we'd like to do with the money. The other half of that equation is getting help to do those things with the money. And so this is the board, this year's board of directors, who, if you're here, please stand up so that we can acknowledge you. But if you would like to be a part of the THS and help us in any capacity, please let us know. Today we have Ryan Perry, Dale Turner, and Cassidy Lee, who are interested in being on the board of directors. And so I will start. They're all here. So I've asked each one of them to stand up and just give us a brief introduction to themselves and why they would like to be on the THS board. So we'll start with Ryan.
So, I'm Ryan Perry. I've been a member for a few years. I started helping out a lot more this last year with a lot of outreach events. I'm also a high school teacher, so I plan on use the experience I've done as a teacher, as well as last year with the society to try to do more. Fired up and funded to make this every year. For the early 90s, served on the board for 4 years in the 90s, including year as president. Other conservation things and various bits of research on works and other things for other stuff. But I'm retired now, have a little more time, perhaps. I love listening. Yeah, I gotta do that kind of work.
Robert knows I'm working on a huge desert tunnel proposal right now. I'm really excited about that. And being over to THS and getting to know everyone caring with Robert especially has been really helpful. So I want to give back, especially with outreach. That's kind of my
Thank you very much. Now it's time to start the election of the officers. So the officers are the president, vice president, secretary, and treasurer. For that, I will hand the mic off to Lee Oler.
Thank you, everybody. It's good to see you here, and I'm having a lot of fun tonight, and I hope you are too. On your table, there are 4 pieces of paper, and each paper has a bio of one of the officers on here. Especially that guy with the snake eye. I don't know why he does that. It's just so weird, but he does. Karen has just came on last year, and she's such a wonderful, she's always got a good sense of humor. I love that. So if you hear a lot of racket somewhere, it's probably Karen and me laughing about something. And then Patrick, I don't get to see too often, but I know how much work he does, and it's wonderful. And then Maggie, she's the one that cracks the whip over us and makes us behave. And she also keeps track of our money, which is very important. So I will remind you, I'm of the age where I can do get this right, minimum yearly something. And KUAT keeps talking about it all the time. And if you feel like doing that, you can make it out. You can give some money to the Tucson Herb Society through that process because we are a 501c3. I had to stick that in because I've got that on my kitchen table right now. I'm working on stuff like that. Take a look at these before you vote. It's going to be a really tough vote this year. And I want, I don't see the ballot. Is that the ballot box there? Oh, yeah, I couldn't read it from where I was sitting. So put your ballot in that box. Who hasn't turned in their ballot? And yeah, you bring that around, Robbie. While he's doing that, I want to talk to you about something over here and what Robbie alluded to before. Young Cage was a 2-time president of the Tucson Herb Society. If you've been around for a little while, you know that. And he passed away several years ago, probably 10 years ago. But I managed to get this picture over here, this photograph of this rattlesnake, and I've never seen a picture, a photograph so well in focus throughout the whole picture. So I urge you to take a look at that when you get a chance to bid on all these fine items up here. And Bettina, I don't know, but Bettina's last name is Jones. She made 2 more paintings this year. One is Hamlet in the Desert. Oh, she changed it. Sonora Shakespeare Theater presents Hamlet. But there's a Gila monster holding a skull and he's talking to it, just like in Hamlet. And then the real saguaros over here also. So I thought those were 2 outstanding contributions. Now, how are we doing with that box? Oh, where's Robbie? Where are you?
Right here.
Oh, are we all done with the box? All collected? in process, and it was a very tough election. So I wanted to say a few things about young Cage, and he's the fellow that took all the pictures and the packets that you have for door prizes. And back there, you can see the red boxes that Maggie was talking about. There's yeah, thank you. Red box, that's where the little ornament is in there. So grab one if you want one for your holidays. And I don't think Maggie wants to take them home, so do her a favor. I think, oh, he's not voting, he's getting an ornament. Okay, thank you very much. If anybody has any questions about how to become president, vice president, secretary, or treasurer of the Herb Society, see me and I will tell you how it works. Because I don't want to run an election next year about the incumbents only. So we need some new blood here. Thank you very much.
I've been president for too long. Uh, yeah, well, I joined in the year 2000, which was a long time ago. And I became president, I think in 2012. I don't know. Somebody can correct me if I'm wrong, but I've been president for a long time. So I would love to run against somebody. So before I introduce our speaker, I'd like to show you that I've been able to line up some speakers for most of next year so far. If you have any ideas for speakers, let me know. Like to get these things arranged ahead of time so we're not scrambling. And just like tonight's meeting, we record our presentations and make them available on our YouTube channel. So Thomas Weaver is the Arizona-Sonora Desert Museum curator of herpetology, ichthyology, and invertebrate zoology. I don't think you've been there a year, or how long have you been? 2 years. 2 years. All right. Tom is a wonderful human, and it's been really great to meet him and know him. This picture is, oh yes, dessert would be great, I think, about now. Yes. Yes. Visual and culinary dessert. So if we could have those, if we could have the dessert served, that would be great. This photo is of Tom on our field trip to find Phrynosoma dittmars. And you can see by the look on his face that he's not having a very good day. With one of the rarest lizards in North America in his hand. It was a great trip to the Sierra Alacran, the Scorpion Mountains, where this species was first caught on the Carl Lumholtz expedition many years ago. And so today he will be presenting about Project Obscurus, a recovery project for the New Mexico Ridge-nosed Rattlesnake, Crotalus willardi obscurus. And this photograph was taken by Addie Limeroth, who was on that trip. So I'm going to do some shenanigans here and call up his presentation. When you get to your videos, I'm just going to have to play them as the files.
Is there a clicker?
I just have to use this guy here.
Try to get the dessert served before you get started.
Sure, yeah. Let's see here.
What was that? Hmm. This is the slideshow.
Good start? Yeah. I think they're gonna bring out the dessert for everybody. So I guess I don't get dessert. Well, thank you for having me. Again, my name's Tom Weaver. I am here from the Arizona-Sonora Desert Museum. In January, I think will be my second year here, so I am new to the area. I have done a lot of work in the zoo field. I've worked at a couple different zoos. I started out at the Grand Island Heritage Zoo in Nebraska, which is no longer. But when I started there in college, I went to the University of Northern Colorado and I was working at the Rocky Mountain Raptor Center, and my love was for raptors. And I was like, I'm going to be a falconer. I'm going to continue on with raptors. And then I went to my hometown in Grand Island, Nebraska, and got a job at the zoo there for like $7 an hour. And the reptile keeper quit, and I was handed pretty much the largest collection of reptiles in the state of Nebraska at the time, which wasn't all that big, but we had a really good partnership with Reptile Gardens up in Rapid City, South Dakota, where I did a lot of interning up there. I got to learn all my venomous handling, and I don't know if you guys have ever been to Reptile Gardens. Basically, my first day, we were looking for an Australian brown snake that got loose and was in the building. And we were going out at night with spotlights to look for it. And they did find it the day before they opened, but it was kind of the wild, wild west of reptile keeping, but got to learn a lot of experience there. From there, I moved on to the Lowry Park Zoo in Tampa, Florida, where I became assistant curator there and worked over the same position I kind of have now, reptile and aquatics. And then after that, I went on to the Denver Zoo, where I spent about a 22-year career there. Most of my career there, I worked my way up to curator there. So I was over a tropical discovery building, which had everything from fish to Komodo dragons to a very extensive venomous collection. And my favorite, I did a lot of work with amphibians there. I got to work on a lot of different projects internationally with, yeah.
Did you learn all the different things?
Yeah, we can talk about that later. Yeah, so basically at the Denver Zoo, I did work with a lot of amphibians, and that's where my career kind of focused for the last 22 years. Working with the Panamanian golden frog, the Lake Titicaca frog, the giant Hunan frog from Peru, and also did a big facility for boreal toads there, which is kind of my last thing before I left there. But I had been coming down here chasing rattlesnakes and taking pictures of them. And I kind of said, if I ever get an opportunity down here, I think I've done a lot of work in Denver. I think it's time to move down here. And then as soon as I left Denver, this job came open, and I was like, I kind of have to go for that job. So I am new to the area. I'm by no means a Southwest expert, but what I'm going to kind of show you today is our programs at the museum, give a little enlightenment onto what we do in zoological facilities or AZA facilities, how we got involved with this project, and I'm going to focus more on the captive element of it, or the human care element of it, and what our role is in this program. So I come from the Department of Herpetology, Ichthyology, and Invertebrate Zoology. That is usually broken up into 3 different taxa in zoological facilities. For some reason, all the ectotherms tend to get clumped together. So if you look at what we do at the museum, our department is called the HIS department, which is Herpetology, Ichthyology, and Invertebrate Zoology. So we have about 12 keepers there, and we have a lot of volunteers and everything. So we do do a lot of different types of stuff. One day we could be working in stingray exhibit, or you could be catching a rattlesnake out on property. The one thing that I have realized moving down here is that the Arizona-Sonora Desert Museum, people here are very proud of this museum. And it was something that I was I thought was very cool when I moved down here, because everybody in town knows the museum. I don't know if they drive by the sign every day or what it is, but it's like everybody's very proud of the museum. And if you say you work there, they're like, oh, okay, you're good. That's an important role there. The one thing that I really did like about working at the museum, I was always working with international collections. And the one thing that I found very appealing to the museum was our regional focus. And I had done a lot in my career to the point where I was ready to kind of focus. I do miss my trips to South America, but Mexico is just as good. And I've done a lot of work and a lot of trips to Mexico and learned from you guys how stuff works down here. The biggest thing I kind of want to focus on is what are zoological facilities' roles in conservation. When I started my career, the conservation word was just starting to get popular. Zoos didn't really have, we talked about endangered species, we had rare species. Are we doing anything with the species? We weren't really walking the walk. We were talking about it. And what I've seen in my career, that conservation word become more of the role of the zoo as far as in education, as far as how many people that we reach out to, and our collaboration with academics, and different facilities and different government agencies, to where now I'm seeing more of a trend to where you can't really do much in a zoo if you don't have the word conservation. And I think a lot of efforts and missions are going towards, we realize that we can bring in animals into human care, but if we don't protect the wild, it doesn't mean anything. So it's very important that people realize that zoos are a resource, zoological facilities being aquariums, botanical gardens, museums, of course, and the zoo facilities are a huge resource. I started to see departments getting tagged onto the zoos that I worked at. We called them a parasite department, where the conservation department's over there and they don't really do much besides they have their own thing, to where when I started working at the Denver Zoo, the first day I started, I told them that I really wanted to work with the Panamanian golden frog because I've been to some talks on it. And within that first year, I was down in Panama catching frogs. So it was and that's, every zoo has different models. The zoo I worked at in Florida, the Lowry Park Zoo, they were pretty much like, yeah, we support conservation, but we don't support our staff doing conservation, which was kind of a turnoff for me. And then when I started at Denver, I got to do a lot of really cool stuff. So some of the stuff, and that's where I'm going to go into the museum and some of our programs that we have before I get to talking about the Obscurus program. The slide here is called the Olson Building. This is kind of our headquarters at the museum. This is where his department pretty much stays during the day. Our desk, our offices are in there, but we are an outdoor museum. I guess you could call it a live animal museum. I kind of look at it more like a wildlife park. We have about 100 acres. 20% of that is pretty much established as exhibits and buildings. But we have a whole desert out behind us that eventually I'd like to see us do more internal research on property. There's stuff out there. I mean, we've seen tortoises out there that have come up that we've found that we're like, we did not put this tortoise out here. And we don't think anybody else did, but this tortoise has probably been on property for a long time. So there's a definite aspect to it about working in situ, I guess, if you wanted to say that, with a lot of our exhibit, which also brings a lot of challenges. I'm very much used to a rainforest that's built indoors in the middle of Denver, to where everything is controlled, every temperature, every climate, every humidity is controlled. Ours, working outdoors and having outdoor exhibits, has a few challenges to it that I did not quite understand. I did do a lot in Florida with that, but I think the desert is a very harsh place for a lot of animals, and it's something to where, you know, when I was starting, I was like, oh, we can get coach whips in with those lizards and they'll eat them all if we don't do something about it. Or, oh, we have chuckwallas with mites on them. And I'm like, I've never seen a mite in 20 years in my reptile collections. And it's something that we have to deal with being able to do what we do with having exhibits outdoors. And that's one thing that the museum is very much known for, is our outdoor exhibits. And it was one of the first places to be able to do habitats. Through the evolution of zoos, it used to be cages, turned into a containment, turned into a habitat. And now we refer to them as habitats. It is something that's more natural for the animal, and we're learning the benefits of that. Sometimes it's not. Sometimes I feel like we do it for the humans to make it look realistic, but it's not any different than something that we do, you know, in a laboratory type setting. But that's the biggest goal for me, is to try and make that combination work, so that we can display animals in a natural type setting. I think the museum is a good place for people to come out to enjoy the desert in a safe place. We do remove our rattlesnakes off property. I can talk to you about that later. We caught over 150 rattlesnakes last year on property that we have tagged. We paint the rattles and we re-release them. We get about 35% recapture. So there's a lot of stuff going on that you know, I didn't realize working in situ in the wild in a zoological setting. So here's the Olson building. This was built in the early 90s. This is where we all kind of go to work. If you go to the backyards, if you haven't been on a tour out there, come talk to me. Not all at once. We'll see if we can get you out there. A lot of different rooms for different types of specimens. We have a lot of different snake species, lizard species, turtles, you know, aquatic systems, and also a lot of insects. When you're developing a collection plan for a zoological facility like this, you really have to look at, what are you doing? Why are you breeding that species? Is there homes for that species? And I think the biggest part with zoological facilities is when we get encroached into species that need help, that we do need to bring them in for an assurance population. Is there enough space? The biggest thing that I can say from working with a lot of amphibians, you know, 2008 was kind of the year of the frog for AZA, where we were putting a lot of efforts towards amphibian conservation. And with the global amphibian crisis, we were looking at probably 30 to 40% of all of our amphibians go extinct or in major decline within the next 50 to 100 years. There's not enough space. If you take all the space of botanical gardens, museums, aquariums, and zoos, we could maybe conserve or preserve maybe 1 to 5% of that, but it's just not a realistic perspective. And that goes into the ARC type mentality. Are we ARCs? Are we going to save animals from extinction by bringing them into our zoological facilities? And we all kind of agree that that's not necessarily something that we can achieve. It's something that we need to also put efforts into the wild. And that's where you're seeing conservation departments and zoos start to really get bigger and build up and really start to support a lot of different kinds of research. And like I said, the museum being outdoors, a good example of when I worked at the Denver Zoo, I wanted to build a boreal toad breeding facility because we were working really closely with the Colorado Division of Wildlife and the Utah Division of Wildlife. And it took about 10 years for me to get about $35,000 to build a facility, which was built a couple years ago, and now it's producing toads that we're putting back into the wild. When I started at the museum, I joined and I started looking at this stuff, and I was like, we already have multiple programs going on here. The museum has always been heavily involved with different, government agencies and University of Arizona to be able to work on conservations collaboratively. This is some of the outdoor pods that you see for our San Esteban chuckwallas. I do have a lot of people from other zoos ask us how we're breeding our chuckwallas, and I say, we put them outside. Very naturalistic. We do have hibernaculums for them that right now we're starting to really look at the temperatures close because we're starting to get temperatures dropping. We probably have about over 80 chuckwalla at the museum right now. I'm trying very hard to move some of those animals out to other facilities. so we don't have so many at the museum. And then some other back areas at the museum where we have a lot of our insects. Again, this is our natural habitat for the chuckwaws. That is a hibernaculum in the middle that we just went through our heating pads in there to make sure they're on and running. And then fish. We have a lot of aquatic programs going on here. So this was a facility built outside of the Olson building to be able to house many different species of native fish. I will go down the list of some of our projects that we have going on. This is one of my favorite slides. When I was younger and I was working at the Denver Zoo, I used to argue with my boss about zoos don't really do conservation. AZA zoological facilities don't even compare to other agencies like the Audubon Society or the World Wildlife Fund and stuff like that. And going and doing some research on that, you can take a look here that about 208, this is AZA zoos. When you walk into a zoo, you look at the front door should have an AZA sticker on it. That is different from other facilities. Like, I don't want to put any down any other facilities, but we're not the Tiger King by any means. If you look at what we get accredited every 5 years, and when they come through, they look at everything as far as our facilities, our finance, our merchandise, our pest control, our sustainability. So we go through, we are going through 2025. We're going to have an inspection this year. And they look at our conservation programs and what we're doing. As far as the statistics here, about $208 million spent on field conservation, which is affecting right around 1,000 species and subspecies that have benefited from that. And that has ranged pretty much about 115 countries worldwide. And the biggest, the one at the end here is probably my favorite. According to the Association of Zoos and Aquariums, over 181 million people visited U.S. zoos and aquariums. It accredits every year, which is more than the NFL, NHL, NBA, and baseball combined. Globally, 700 million people visited zoos and aquariums every year. That's about 10% of the world's population. So I guess the biggest part of that is our awareness that we can bring up with people, the amount of people that we do touch that come to our museum to learn about wildlife. So that is one of the things that why I work in a zoological facility that's AZA accredited. Some of the projects that I'm going to go down a list. This is a Mexican garter snake project that we have a group at the museum that we bred every year and release offspring. Stephen, how many did we release this year? 47 babies this year. We were getting ready to go out, and we had to stop the trip, and we had about 30 babies born the next day, and we're like, well, let's hold off. Let's wait until we get these babies a little bit bigger, and then we went ahead and released those. And then, of course, our tortoise program. Rachel's here. You can talk to about a tortoise. We have some of the museum staff here. We do have a tortoise adoption program where we do bring in tortoises. We'll take in tortoises and find good homes for them. This program is a very big part of our whole operation. It's basically a keeper effort throughout the entire summer seasons, to where we Rachel works very hard at talking to people on the phone. We go out and inspect people that want to get a tortoise, make sure they have the right facilities for it. And we do find homes for a lot of tortoises, and there's always tortoises to find homes for. Even if we run out at the museum, we have about, I don't know, I know the state usually has about 300 tortoises they're looking for homes for too. Sonoran mud turtle is another program, which hopefully next year we'll be releasing some babies. I always like that M&M photo of how big they are when they're born. Pretty cute. Fish, we do a lot of native fish species. We do work with a lot of chub species. I'm learning all my native species. And we'll go through some of those with the list. Amphibians, we do work with the Tarahumara frog. There's a Cherokeensis frog there right in the middle. We aren't working with Cherokeensis right now. but I'm looking at potentially getting us involved in the future. And then one of our biggest programs that I talked about was our San Esteban Chuckwalla program, where we have probably over 80 specimens right now that we are trying. This is more of an SSP program, a species survival program by AZA, where we work collectively with other zoological facilities to make sure that we are not inbreeding our animals and that we have a healthy population for the next potential 100 years or so. So here's some of the projects that we are actively or have historically worked on. I already talked about most of the ones on the left, but on the right, Keto Vaquito snail, we just kind of finished up that project. The snails went back to Phoenix Zoo, which they are working with. Rio Sonoida pupfish, Rio Sonoida longfish dace, Gila top minnow, Sonora chub, Yaqui chub. So we do have a lot of programs in the works. The one you see on the left-hand side, the new one, the New Mexico Ridge-nosed Rattlesnake. So that's where we got started with this. When I started 2 years ago, my first meeting with my boss was, we are involved with this rattlesnake project and it's your highest priority for the next 2 years. So basically I was lucked into it. I got involved with it right away. We started planning for how we were going to help assist with this project. The funny thing about this, knowing that this trip was a year and a half away, I got calls from all my rattlesnake friends that wanted a picture of an Obscurus in the wild. And I had to tell multiple people that I am not in a position where I can invite people. We are getting invited ourselves. So there's a lot of people out there that have taken pictures of all species in North America that don't have this species yet. So I thought that was kind of funny. To go down through the project, what I'm going to show you, I'm going to go through, I'm going to show you a video. We did do a short documentary that was very good documentary that I felt like I don't like to do this in my talks to do a big video, but I figured there's no reason why I shouldn't display it here. But if you want to get a good look of willardi and the Obscurus range, if you look at the top of that slide, you see the red dots there. That is where Obscurus historically has been. And this is a kind of a close-up look at it. If you look, I don't really have a pointer, do I?
Is that's California on the last picture, right?
Yeah. Yep. If you want, well, I'll answer your questions after. Okay. Pretty much when I get done with the talk. Yeah. So here we go. So if you look up through here, you can see where the historical range of Obscurus was. I believe these are the Plencios and the Animas and the San Luis Mountains. Just to get a good idea, I don't like to put localities on my presentations, but everything I looked up today had the locality on it, so I didn't feel guilty about putting them up there. The topography of the habitat you're going to find, this is the Animas Mountains, very rugged, about probably around 7,000 to 8,000 feet in elevation. Very hard habitat to survey. And I'll talk a little bit more of that when we get going on the project. Rough terrain, montane terrain where the different types of habitats. And then we'll go down through the history here of the program. Like I said, I'm not an expert on Obscurus, and I'm sure there's some people in the room that know more than I do that can answer questions probably a little bit better. But basically, the first specimens collected was in 1957. I was trying to figure out when the species was first described, and it seemed like there was some controversy on the first description of it, as far as the taxonomic nomenclature and as far as how it was described. But I think it was described in 76 as Obscurus. And then in 91, Barker and Painter did some investigation, natural history of the ridge-nosed rattlesnake, and then basically through here, it ended up in the biology of the pit vipers. Surveys have been going on since 1990, all the way up to 2024. And then we can go ahead and get started this. We'll go ahead and start this project. Oh, here we go.
We are undertaking the largest assisted migration project.
So this is about 8 minutes, so we'll go ahead and go through this. We're trying to get sound here.
Worldwide. It's also the first real recovery action for this endangered species. The mission of the Rattlesnake Conservancy is to conserve rattlesnakes and their habitat, through research and education. Our team with the Rattlesnake Conservancy are presently in Sierra San Luis in Mexico for the New Mexico Ridge-nosed Rattlesnake, Crotalus willardi Obscurus. It's a multi-agency, multi-partner involved effort. Andy approached me about this project. He's been working with this species forever, a leading expert on Obscurus in the United States. And we got to talking about this project, and a few months later, an opportunity came up to apply for the Recovery Challenge grant and got it submitted. About a year later, we found out that we won the Recovery Challenge grant, and this was a game changer for our organization to have funding to pursue an active conservation project for rattlesnakes and be able to make a difference for these species.
I've been working for the service for about 8 years now, work out of the Albuquerque, New Mexico Ecological Services Field Office. So I'm the lead for a number of amphibian and reptiles. So the New Mexico Ridge-nosed Rattlesnake is a species that was listed quite a long time ago. It's like 1978. The conversation became, what could we do? I worked with the species expert. They drafted a proposal. We applied for the grant with their Rattlesnake Conservancy tail. And then the Rattlesnake Conservancy and the research partner were able to get it. So now we're here and we're going to be releasing rattlesnakes.
There's a number of different objectives with the Recovery Challenge Award. First and foremost, we're trying to get 30 animals into captivity with 3 partner zoos. We've got LA Zoo, we've got Arizona-Sonora Desert Museum, and we've got San Antonio Zoo. We hope to get 5 males and 5 females to each of those institutions so that they can experiment with captive breeding.
One of our specialties is mountain rattlesnakes. We work with a lot of Mexican mountain rattlesnakes, including the New Mexico Ridge-nosed Rattlesnake. It's kind of what we've been doing for 2.5 decades now. Oftentimes in zoos, some of these obscure species are overlooked a little bit. We ended up being the first successful zoo with New Mexico Ridge-nosed Rattlesnakes, and we've just been taken off from there.
We focus on basically taking care of animals that are representing animals that are from our region. We are involved with this program to be able to bring in animals, wild-caught animals, into the museum, basically to reproduce these animals to put them back into the wild.
We're really excited to be working with Obscurus, bringing in the New Mexico Ridge-nosed Rattlesnake and adding this program, something that all of Los Angeles Zoo is really proud to be participating in.
It's really great to see everyone working together to save this species.
Second component of the project is genetic augmentation of a U.S. population, the Palencia population. We're hoping to bring 12 animals into the Palencios in hopes that a couple of those survive and contribute genes to that population, which is genetically impoverished.
For the project as a whole, our role has been relatively peripheral, but because we're releasing snakes into Arizona, we have to take a central role in that because only the Game and Fish Department has the authority to release animals into the wild. But I've been associated with many of the researchers involved in this project for many years. This snake has been listed since 1978, so we're going on nearly 50 years before any kind of recovery actions have taken place. What we're trying to do is basically genetic rescue for the animals in the Palencia Mountains by bringing in animals from the Sierra San Luis. The idea is that they're going to infuse fresh genes, if you will, into the population and perhaps make that population more resilient to environmental changes that might take place over the next 100 years. It's truly an experiment, and I see this as a very important step forward into actually trying to do something for the recovery of this particular species.
Third component of the project is we're hoping to get temperature transponders in 8 adult males and release them to the wild. Those transponders will record temperature every 5 minutes for a year. So we'll know exactly what the thermal requirements of the snakes are. And that'll be helpful both for the zoos with their captive care and also to some habitat modeling that we're doing with USGS partner.
Alright, snake number 3 is awake and alert after surgery. Snake number 130 fully awake.
The final component is that we're using operative temperature models, 300 of those, to try and quantify the thermal landscape that's available to the snakes in these mountain ranges, and that's also a critical part of our habitat modeling.
I've been working with willardi obscurus for about 32 years now, working with the thermal biology of Obscurus. Climate change is affecting the Sky Islands in the southwest US, primarily in New Mexico and Arizona, and in New Mexico for this species range. Because of climate change, we're seeing a drying trend in these mountain ranges where this species occurs. No one's done a lot of thermal work with this species to look at how climate change is going to affect their future. This project is a huge endeavor for a species that most people don't really care about because it is a rattlesnake. If you remove that species from that ecosystem, you're going to have some sort of effect.
As far as the species goes, I think that Obscurus, or any species, is sort of the distillation of a time and place into an organism. Obscurus in the Palanceos and the Animus and the Cirrus San Luis is that ecosystem in the form of a little snake. And if it's lost from that ecosystem, then the ecosystem is now incomplete. As I was studying the effects of climate change and fire, it got really, really depressing, especially in 2021 when a fire burned down my study site. After that, I got the feeling that I wasn't really doing active conservation as much as I was just watching the end of the world very, very closely as it affected this one spot and these couple species of snakes, Obscurus specifically. I remember thinking to myself, if only there was some sort of conservation effort, if only we were actively putting snakes out on the landscape or breeding them, anything. And then I get the email to do this, and it's fantastic to know that I'm not the only person who was thinking that.
Relating to this project, this is I think that a pioneer effort on conservation of rattlesnakes, because I think that it's going to pave the way for many people that wants to do something similar with other vipers in other parts of the world.
One of the most important things about this project is its collaborative nature. This would not have come together were it not for the efforts of our colleagues in Mexico, you folks at the Rattlesnake Conservancy, Andy Holycross's initial impetus from the academic standpoint, the U.S. Fish and Wildlife Service stepping up, Game and Fish playing a role, and this whole array of volunteers and zoos that are participating in this project. This is almost unprecedented when it comes to amphibian and reptile conservation.
43 people came together in this camp out in the mountains every day looking for this species and really targeting their efforts to help find these, to help recover an endangered species. Everyone here is like-minded in that they want to see this species succeed in the wild and recover.
I've never seen more people come together and really just put aside any differences. And it's beautiful to see something like this occur for a species that most people wouldn't even know exists.
We're all working together to try and see this through and see it to a successful conclusion.
Yeah.
Okay, well, thank you. That turned out really good. We're very happy with that. Thanks to the Rattlesnake Conservancy for putting that together. So to go back on the goals of what this recovery challenge was to prepare basically to prepare the possibility of assisted migration. One, to assess 3 captive breeding populations, Los Angeles Zoo, San Antonio Zoo, and at the Arizona-Sonora Desert Museum. And then develop more refined predictive habitat model, examine relationships between thermal environment and distribution of New Mexico's ridge-nosed rattlesnakes using 350 operative temperature models, quantify and describe dealing with seasonal variability of body temperatures of free-ranging ridge-nosed rattlesnakes. So this trip was quite the trip. I've done a lot of work in herpetology. I've done a lot of herping. I've been throughout the Amazon and through a lot of different cloud forests in Central and South America. And this was probably the most intense trip I've done ever, really. We would get up in the morning about 7:38, and we would hike all day long until it got dark. And we had 47 people that were intense, like snake hunters. So it was pretty cool to be able to be out with those many people. And the habitat that we were going around was basically, we had enough people that every 10, 15, 20 minutes, somebody else was. So if you walked by a snake and missed it or didn't see it, somebody else was walking over it pretty soon thereafter you. So, and I think the first day we had caught 3, and we were like, we're screwed, we're not going to do it. And we were talking about, well, maybe we should come back in October. That night it rained very hard, and then we woke up in the morning and it got really sunny and warm. And that's when we started really collecting or finding a lot of specimens. So to go back to the museum here, to start off with our captive program, like every zoological facility, we have exhibits. This is one of our areas. We're right now in master planning to develop a new herpetology facility within the next 5 to 10 years. I'd like it to be more 5 than 10. We do have a lot of exhibits. We do exhibit majority of all of our stuff, or all of our stuff is Arizona or Sonora species, focusing primarily on the Sonoran Desert. Our Montanes is one of my favorite exhibits that we have, where we already had Obscurus on exhibit, where we have a Lepidus on there, and then we have a price eye on there. But to basically focus to show the species that we find in the state of Arizona or in the region. There's a good look of a Obscurus right there. And then we also have different species up there, like your pretty much your Southwest exhibit tree, where you find all the rock stars of the Southwest. Melosus there, and then Lepidus. If you go into our Olson building and look, we have a lot of different space. We do have space to be able to do an isolated quarantine here. We did agree with the program that we were not going to focus on isolated quarantine, because the other facilities, like I was talking about with space, do not have space to be able to isolate their rooms. We fortunately do at the museum. We have our garter snakes, or Mexican garter snakes, are isolated. The willardi or the Obscurus are isolated in a quarantine facility. This is the room that we decided to use. If you look at that snake rack, we don't really use snake racks much anymore. We try to have most of our stuff in the back for tours have open. We're using vision cages to be able to look into them and be able to have light on them. This rack that was built, I don't know, probably 20 years ago, was built out of cutting board material by my boss, Stefan. And it's the heaviest rack I've ever tried to move. Although it's still standing, is very strong. What we decided to do, we put all the adults into vision cages, and I'll go through a little bit about what type of material we use for them. These are in a room where it's shut off from the rest of the collection. All the tools, everything is found in this room. We don't move tools from different rooms. What we wanted to do is we wanted to create a little habitat where we have substrate that also is able to hold some moisture. Cypress mulch, or just mulch in general, is probably going to be too dry. So we went with a combination of different materials that we got from the San Antonio Zoo. But we wanted to give them the ability to go ahead and bask, to regulate their temperature, go hide in their hide box or go under the cork bark. The rack system that we used here, these glass containers are actually, I think, coral reef fragment type tanks that I bought. And they actually worked out really good for venomous. So what we did is we split the racks down to half and half, and then we have lighting on one part of the rack that is able to warm up half of the tanks and then cool down and keep the other side basking spot so that snakes can thermoregulate between both sides. And if you look also, like everything that we have venomous, this isn't a locking room. We all have our individual key for the locking room. So we decided not to put venomous locks on all the species or all the different individual tanks. And this is basically worked out pretty good. We've been seeing some good growth and some good behaviors from these species, being able to thermoregulate and move around the tank. There's one right in the middle. You can see them hanging out on top.
So does each habitat have a different controller?
Right there. No, they're all on the same controller. We basically monitor them the same way, and I'll get a little bit more into that with our light cycle. The one thing that they did really well with the Olson building is the rooms are very much conditioned to the outside. So a lot of the focal or the photo period and everything like that is all natural. We do have UV transparent glass coming in through one side of our buildings for all the species. It does work kind of if you keep it clean. You can get about 30 to 40 reading on the UV meter. The combination of what we did for substrate was mulch. Obviously the cork bark here, which we stacked in there, we use just basically cypress mulch mixed with, this is called an ABG material, which is Atlanta Botanical Gardens material that was designed there. I was kind of helped with that too, with a lot of, that's more of an amphibian substrate that's used a lot. And I think it came from a botanist who worked there, Ron Gagliardo, who developed that. And then sphagnum moss mixed with some leaf litter. So again, we didn't want them. The one thing we did find when we did bring them in, they were really dehydrated, and we had to really look at. We did get some real small ones that were under 5 grams that we were really concerned with. Probably too small. Probably, I think if we did it again, we wouldn't collect that small of a specimen. I don't think those specimens. I think they were just born. They were neonates. They hadn't shed yet. So basically the setups, we do have water bowls, but I'll show you here in a little bit why we probably don't need water bowls with them. See how they kind of utilize that cork bark, and we'll go up. You'll see them throughout the day sit up on top, and then you'll come back in the afternoon and they'll go back down and kind of cool down. This is kind of a photo period that we were given from San Antonio Zoo. We're trying to replicate this as much as possible. We're kind of behind. Right now, what we're doing is we're starting to cool the room down, but we're going to put them through that photo period, the natural photo period, and bring it down to right around 55 degrees for their cycle throughout the winter. Again, like I said, the room is, all the rooms were made with sinks in them. We have our own hose bib. We have all of our own tools in there. And if you look in here, that's food. We learned very quickly with some of the little ones that we had, they were not eating prey that we were trying to feed them off hemostats with pinkies, parts of pinkies. One of them was 4 grams that I was getting very concerned about. I ended up force feeding a baby that was 4 grams 2 or 3 times before we started feeding them centipedes. That was very, it was a lot of pressure. It was like, give us something, just give us something that we can keep alive and not have to really risk trying to get it into a rehab situation. But I was able to force feed it and get some food down it. And then once we started feeding it centipedes, it took off pretty quickly. These are centipedes that we brought in that we have in the room. The funny thing about this was, so we were collecting centipedes, I ordered centipedes. Once we got them to eat centipedes, we realized, okay, we're gonna have to do this next week, exactly the same. When you feed, I've worked with saw-scaled vipers at Denver that will eat scorpions. But I think I didn't realize with insectivore neonates, they have to eat a lot. If you feed it a pinky, you got a week to 2 weeks before you need to feed it again. But if you're feeding it insects, it's gonna go through them right away. So it's a lot more effort. But once we did feed them with the centipede, it was an innate response. It was very, very quick. Tongue flick, and then they grabbed it. And that was when we were like, this is awesome. Now that we have the scent, not necessarily the centipedes, we have the scent to be able to work with, to be able to get some of the other ones to be able to eat if we have trouble with them. I think one of our female that was pregnant that delivered babies, we fed her, she started on a quill. And part of their diet we'll go through here. If you look at, these are studies that were done on the type of diet that pretty much willardi or obscurus have. It does have centipedes, lizards, mammals, and birds. My old boss, Dr. Charlie Radcliffe, was collected. He had probably the largest collection of rattlesnakes in the 70s. He was up in Boulder, Colorado. He remembered, he told me, after I got back from this trip, I told him that we got him. He goes, I remember I had an obscurus that the first feces I got from it had warbler feathers in it. So that was kind of a key to us. So a lot of these experiences, a lot of the literature and a lot of the studies done on is how we decided on how we were going to approach trying to feed these animals. It's another statistic there. I don't know if it's covered up the top. But yeah, the centipede thing was very exciting to us because I've never seen a species go so quickly for a scent item. The neonates that we did have a gravid female that had 6 babies. We brought in, what was it, 10 specimens. We tried to get 5.5. We ended up getting another male from the Palencia Mountains to add to the bloodline with that. But we did, I mean, at the museum, I've learned a lot about, I've done a lot with trying to raise baby pit vipers, not necessarily rattlesnakes, but I did a lot of work with Asian pit vipers at Denver. And it was kind of my thing to be able to get them to eat. I was always the one that's like, let me have them, I'll see if I can get them to eat. But these guys, as far as the centipede goes, it was pretty exciting to see. And you can see babies, all of our babies were right around 8 grams. So they are little guys. What we ended up doing with the babies that were just born, we let them go through their shed, and then we ended up taking a centipede, sorry to the entomologist, but I froze a centipede, chopped it up, and made a little slurry out of it. So it's kind of like a centipede shake. And then we soaked little pinkies in that to let them thaw out, and they pretty much, after a tongue flick, took the prey item. Just more pictures of babies. Take a look here. These are the ones that were just born. You can see by the eye turning blue there. This is prior to their first shed. I got another video here. Let's see. So there's no sound to this. I'd like to credit Stephen Comerworth here as one of our keepers that did pretty much all this footage. A lot of the pictures and stuff here came from Andy Holycross, and some of them were mine, but a lot of them was Stephen's daily observations and spending time with these animals to get some footage like this. So here's eating a centipede. The story behind eating the centipedes, we went ahead and collected some polymorpha, and I threw them in there to see what they would do. And the first thing they did, they attacked the snake, and I was like, this is not what we want. And trying to get a centipede off a snake with hemostats was kind of difficult. So what I ended up doing, or Steven, we started doing as keeper staff, was cool them down, the centipedes down to almost where they're frozen, and then let them kind of go after them that way, so they're kind of slowed down. You can see that centipede is still kind of moving his back legs, but still that was kind of dangerous. I ended up chopping a head off a centipede and throwing it in there to see what it would do, and the centipede crawled around all day long without a head. And we left it in there, and we came back in the morning, and the snake had eaten it. This snake here is probably right around 5 grams, so you can see how big that is. There we go. Did we go back to that?
You're drinking?
There was one we missed.
Oh.
I go back. We got another video that I forgot to show. See if you can click out it. Go back.
This one?
Yeah. So here's why I keep joking about water bowls. When you work in a zoo, you have a lot of animals, especially in the reptile department, that probably don't know what a water bowl is. Never seen one. I mean, if they come across it and they mistakenly touch their mouth to it, they probably will notice it's water and they will drink about it. But I realize working in a zoo, you have to have water with everything. You have to make sure everybody can see that there's a water source. Here's a morning where there's mom behind there with all of her babies, that we just went ahead and missed it, and you can see them drinking droplets off their backs. And that became very acute to us. It's very important that we do at least one day misting in the mornings with them. Some of the questions I got brought up is, do you keep mom with the babies? I don't like to anthropomorphize animals. Whenever I work with keeper staff, I try to teach them or try to understand that we have to learn the animal's language. You have to throw out kind of your human perspective or anthropomorphosis and like think like that animal. If you come in the morning, like I was saying, like when I worked in Denver, a green tree python is sitting on the ground every day, all day long. To the keeper, he has to think like, or they have to think that maybe something's going on. Maybe we need to check our basking temps. Maybe we need to look at our overall temperature. Maybe we need to do a fecal on the animal, see if it's having a parasite load to it. But stuff like this is where, as zookeepers, going into the wild and working with the animals and bringing that back to the zoo or the museum to be able to apply that knowledge, in human care is very important, I think. But one of the things my boss told me is I don't know if they need to be with their mothers. I know with a lot of the rattlesnake roundups and stuff, there's a lot of footage from the Dallas Zoo that had moms, in the rattlesnake den with moms sitting with a baby, the baby sitting on it. We have a rattlesnake cam that's off for the season that we have posted in our reptile exhibits, where you see babies coming out and sitting on the adults. And as far as I look at it, it's like, okay, that's fine. If you correlate a human behavior or correlate moms and grandmothers and aunts and uncles living with their babies, that draws a connection to the human. And I think that's, even though us as scientists may really question that and look at, is that something that is needed? I don't know. A lot of the times there's been a couple observations at the museum where we've had mother atrox, have a litter of babies, and mom would leave and come back to them. And we don't know what she was doing. If anybody else here has more knowledge on that behavior with rattlesnakes, I'd like to talk to you more about it. Some question that moms will go about and eat and come back, and the babies will tongue flick their mouth to be able to get a cue on the scent. I don't know how that applies with the centipede, because I don't think mom is eating centipedes, but she might be. I think from what we've seen at the museum, these babies are getting born, they're going through their first shed, and they're going through the bottom humus layer of the forest, and they're coming upon centipedes. And they're nailing them as soon as they get them, they're envenomating them, then they go ahead and eat them. The interesting thing about the funny story about the centipedes is we had a bunch of baby polymorpha in with the rattlesnakes in the same room, and one of the keepers walked in and goes, "Man, it really stinks in here." And I was like, "I don't know what it is." I go, "It's not the snakes." I go, she goes, "It's the centipedes. It smells like the centipedes." And I was like, I didn't know centipedes had a scent. But I started noticing it, and then I went on my weekend and I came back and I was checking on the animals, and I looked in the sink, and there's a giant desert centipede hero in the sink that couldn't get out of the sink. So I called the keeper staff right away, and I was like, "What's going on? Did somebody collect this? You think we're going to try and feed this out to the baby snakes?" And everybody on staff was like, "We have no idea what you're talking about. Nobody brought that down. We don't know anything about that." And so I started looking at a trail of where this animal might have come from. And of course, down the hallway a little bit, we had a little crack in the bottom of the door to the outside, which I'm sure mice are getting in. I think what he did is he came in, he scented those other centipedes, and ended up in the sink, which is very ironic. Talking with some of the other entomologists or the private dealers that are selling stuff, they're like, every time I go look for centipedes, I usually find a big hero along with the other ones. So interesting, but it was kind of funny because I thought somebody was messing with me, because I was like, "There's no way this thing came up through the sink. It's too big." But now we're winding down to the end of the talk. I'd like to thank everybody that helped with this program. We'll go back to that a little bit. This is more from Andy's slides. A lot of different, the one thing about the collaborative effort with this was there's a lot of people involved, a lot of organizations involved. It was very important that we make sure everybody gets basically notice on if they were involved with this program. I had to use that picture somehow. So I did a little joke about volunteers. Volunteers were very core to this program, especially when it was out in the field collecting. Where we went was very wild. We had bears on property, we had mountain lions, we had bears scratching at tents in the night. And it was definitely one of those things where everybody got along, everybody there was for the same mission, and it was a pretty incredible experience to be able to be involved with that. And then some more acknowledgments, our volunteers, of course. Talk about everybody involved with that. So thank you very much. I appreciate you having me here. If you guys have any questions, I can try and answer them as best I can. All right, now for questions, go ahead. All right, bro. What's that?
Shape made of a snake? No.
No. When we're I call it the herpetologist bag of tricks. Usually, like when I started as a herpetologist, I was lucky enough to have green tree python eggs, and I had a bunch of babies. I couldn't get them to eat. I could not figure out how to get them to eat. One of the older keepers came by and he goes, "Let me try." In 45 minutes, he got like 10 of them to eat. I was like, "What did you do?" And he showed me how to do it. But as you gain experience and you work with a lot of different species, you learn how to get those animals to feed. And a lot of it is letting them bite the specimen or the mouse or whatever, the pinky, and then freezing and backing off and not move and not distract them. And I think a lot of times when you're feeding different snakes, they go through they get tired and they get that in their mouth. And if you kind of back off and let them hold it in their mouth, they wake up and they're kind of like, "Oh, I guess I should eat this now." I've done that with many different species, but utilizing those tools to be able to get these guys to eat was very rewarding once we got them to go. So we're doing a lot of I've done, at least in the last 2 years, I've tried to get a lot of different species to acclimate to captivity. We go out a lot and catch a lot of lizards on property. We always have our lizard noose in the car, ready to go, to be able to get food for a lot of the tough feeders.
So how'd you get them to eat?
Basically, you tease feed them, they grab onto the food and you let them go, and then they kind of wake up from their torpor a little bit, and then they go ahead and eat it. Anybody else have any questions? Go ahead over here.
I think drives the difference in phenotype between Obscurus and the other willardi population.
Drives the phenotype. I don't know the answer to that question. I would say, I don't know. I mean, they definitely are different looking. I think the Obscurus, before I started this, was probably the ugliest of all the, I think, 5 subspecies, or at least historically. But now I don't think that. They are, I don't know. That's a good question. Does anybody else have any answers for that question?
So more sturdy. If they look.
Go ahead, Tom. I knew Tom was going to be able here to help me. Basically, when we were feeding them cooled down, but they do envenomate them. They do strike them and let go of them, and then I think they kind of follow them around the scent. Yeah, I think what they're doing is they're cruising around under the humus layer, and if they smell them or touch them, they're going to grab onto them. And we've had ones eat them from the side, and I've seen them kind of get them in their mouth. I mean, usually when you feed a prey item to a snake, they will seek, if it's a mammal feeder, it's going to seek that nose or the hair membrane, or the whiskers on it. These guys will eat them basically any way they grab them. They'll just go ahead and start to eat them. Yeah.
They had baby snakes and they were killing them and eating them.
Yeah, I have a lot of respect for centipedes after this. And when you pick up a rock and you see a centipede and you try to put it back really lightly, don't worry about it. They survive. I've stepped on them and they didn't hurt them. Go ahead.
So put those in animals that were left in facilities, or just animals that...
Yeah, we weren't really a part of that when we got set up at the camp. There was a couple of houses. We were all in tents. Well, most of us were in tents. They were in the house and they didn't want anybody in there. They were working with a lot of venomous stuff, and they did not want to have any accidents happen or to get it more busy. But I do know those will record temperature every 15 minutes for the next year. So that's pretty incredible. More susceptible to climate change? Yeah, I don't know the answer to that question. I think there's a combination of a lot of different things in the United States. As far as I do know, that is a very popular animal that people want to collect. I know that there is that aspect to it. But like we were saying in some of the literature, I think Dr. Holycross refers to it as amount of days it takes for you to find a specimen. And I think in the Palencios, it was like 30 days to be able to find one specimen. In the San Luis, it was 3 days. And like I was saying, the day that we didn't find anything, they were there. I mean, we're walking right by them. It's just a matter of them coming out. So a lot of the research I've done, like with boreal toads, we had 15 people walking for 5 days and we catch like 2 toads. And I know we were walking over a lot of. Go ahead over here.
Are all the rare subspecies of willardi geographically isolated?
I don't know the quick. No.
Are all of the less common subspecies of willardi geographically isolated?
I know in Arizona, historically, we have Obscurus and we have, willardi, but all the other subspecies are from Mexico, I'm pretty sure.
Because they're a different style